So welcome everyone. This is lecture number 8 of computational biology model methods using virtual tissues. We will be talking today about diffusion limited growth, how to combine what we learned last week about diffusion and uptake with what we know already about how to control cell growth and mitosis in order to model what is biologically observed in tissue that cells proliferation is limited due to nutrient availability. And once we're past that topic, we're gonna start a um, somehow related but new topic. We'll be talking about chemotaxis, how cells can move guided by concentrations, by detecting signals and changes in the concentrations of those signals, and how we can model that cell motility and implement it in CompuCell. Just as a quick reminder that the class is being recorded and live streamed through YouTube. So I'll go quickly over this, but this is just a summary of what we'll be talking about today in the lecture. As usual, we'll start with the student project updates. I want to hear what the progress has been in your respective projects. I would say that within the next couple of lectures, we should have covered all the basics of CompuCell plugins and the what available options users have to model different cell behaviors. So you might have already in your you might already know plenty of the CompuCell utilities that probably you will require for your projects. In case we haven't covered something that you come across while you're working on in your projects, please do let us know, and we can include it in future lectures. We will review a little bit of what we talked about last time, how to model diffusion through membranes, what happens to the concentration profiles when we include diffusion and decay, how to represent exocytosis and secretion in CompuCell, and how to represent endocytosis and uptake. And then after the review, we will have a in-class exercise where as I said, we combine the uptake from the concentration field with cell growth to control how cell proliferation or to model how cell proliferation can be controlled due to resource availability. We will talk later about a little bit of the biology behind self-movement due to gradients in different types of cells, and how this and then how this can all be represented and think of as chemotactic movement. Then we'll talk about how chemotaxis is modeled and represented in CompuCell, and we'll play with how the chemotactic force or the movement of cells can be controlled by changing some of the model specification parameters that we already know. And then we will, at the end of the class, we will have 2 exercises that I think are really fun, where we can actually start seeing combining some of the stuff that we already know, especially and that we've been playing with over the last 2 weeks, to construct rather cool and actually quite realistic models of cell behaviors. So, okay, let's start with the a little bit of an overview of the student projects and your updates. I'm gonna start from the bottom of the list up, and it's gonna be Tyler. Just let me know if you need to screen share, so I stop sharing, but it's not required.
Yeah, I have a couple slides to share and some screenshots.
Okay, I'm gonna stop sharing and give the screen back to you.
Okay, so my goals for this week, my goals for each week actually have been changing as I update the elements and add elements to my project. The logical flow of how I'm going to build this changes week by week. But my goals for this week were to implement contact-dependent cell differentiation, begin implementation of cell apoptosis for certain cell types, as well as lifespans for cell types. And I wanted to initialize the cells to be spread out throughout the simulation domain, as opposed to being in one clustered blob. And so far, I have here seen the big red cells are the APC. And when they come in contact with the blue naive cells, they become green, which is pre-activated. And there's the proper number of cells throughout the domain, and they're all spread out. And what I need to work on still is the diffusion field, so that when the concentration of my IL-2 field reaches an appropriate value, these pre-activated green cells will become activated. And I also need to work on differentiation for activated to effector, as well as secretion of the field. And that's pretty much where I am right now.
So I actually have two recommendations. I think it would be useful to start drawing a flow diagram of, now that you've been, because you seem to be putting a bunch of cell behaviors together, which is cool, it's really nice. But so you can keep track and also explain what the model is doing, it would be good to start working also on a flow diagram, just like the flow diagrams we worked with last semester. But in this case, a flow diagram that explains cell type changes, but also what sort of behavior cells might have. And there's a couple of references that I can pass along that have quite nice, or that have very detailed flow diagrams, maybe more detailed than what you need at this point. But yeah, just so you keep in mind what the model is, what are the behaviors and components of the model, and then you can also explain it to other people as well.
Okay.
Okay. Next
Josh,
is Josh,
yeah, go
ahead.
Let me just share one slide. So let me suggest that, and I think, Josh, you have a longer version of this perhaps available to show people, but so this is a possible way of organizing what how you might build a model. So this is a table for a particular model, and it lists a particular set of cell types, and then it lists in the second column what they do. And here we have, for example, neovascular cells, we have vascular cells, we have a chemical field, long diffusing VEGF-A, short diffusing VEGF-A, and so on. And so this is an attempt to take it just in a formal way to organize the information that you're putting together. And there may be other kinds of diagrams you might want, but that might be helpful in terms of a way of presenting this information. I don't know, Josh, what you think about that. I think we actually have a couple of slides that go into a little bit more detail about how to put something like that together.
Yeah. Yeah, I think it would be useful to go over it probably next week. Not because as people progress through their projects, their models become more and more complicated, and then just as a way of keeping track and also presenting the content of the model, I think it becomes easier if the information is organized in a.
And I think it's worth. I understand that it's hard to write a specification for what you're going to do in the following week, but I think it's worth spending a little more time for some people on actually trying to write a description of what you're planning to do. And the reason for that is you could think about it this way. At some point, you're going to have to write a paper, which is going to describe what you did. And it's helpful if you could sort of, if at this point now, you could think about what might go into that paper. And so these self-assignments that I've tried to describe to you, the reason I had so much detail requested in those self-assignments was because I know from experience that when you come to writing the paper, you'll need that information. And so if you can organize yourself and try to put down on paper now, essentially what you're going to need later, it'll make it much easier and more efficient when you do come to write it. And so I think that spending a little bit more time, if you can, on thinking about what, not only I'm going to work on X, but on what X would look like when you've done it, will pay dividends later on when you're trying to organize your results. And I'm happy to talk to people individually about that, but I think it's people have been providing very nice reports to me about what they did the previous week. I think that some people have been doing the level of detail of what they're planning, people are planning for next week is varying a lot. And I would encourage you to put a little more time and effort into thinking about what you're planning for next week. It's not that being generic about it is wrong, but I think you'll find that it helps you to spend just a little more time on that as you go forward.
Okay, I have Callie next on my list.
All right, so I have a few slides and then I have some code. All right, so my update's kind of a two-parter because unfortunately I couldn't make it to class last week. So one of the key components of my project in modeling the growth of the fruiting body of Dictyostelium discoideum is this idea that cyclic AMP affects morphology of the cell, so affects how they interact with each other and move around. So a key component of the project is going to be being able to replicate the cyclic AMP models from a paper that was written before my main paper, but contains the actual model. So the main paper I'm working with didn't necessarily have the differential equations for the cyclic AMP on it, so I referred to a previous one that the author wrote. In the bottom, we see figures from the paper. So these graphs, well, the little graphs in the corners are modeling both the cyclic AMP concentration C and the cell refractoriness over time. And you can see they have two different scales on these graphs. And then they're also modeling the cyclic AMP concentration against the refractoriness, which are the larger graphs that you see here. So then above are my results. I didn't do the fancy scaling where I had one scale on one axis, another scale on another, but you can see that the values are the same. Our cyclic AMP concentration kind of peaks at 1. Our refractoriness values peak at a little less than 3, which is what we expect. And then after plugging in the values from the paper, we see the oscillatory behavior for the values we expect to see it for, and the relay behavior, that's the single peak and then going to 0 for the values that we expect for. So was a success. And then in doing this, this is the antimony string that I'm currently using for the model. Still need some annotations. I found that the value the paper used didn't necessarily line up or weren't necessarily on the scale that I wanted. So when I initially simulated the models over here, I originally had like a lot of oscillations in the 200-second window. And I found that the 2 oscillations that we wanted to replicate this plot occurred in like the first 20 seconds. So I found that if you scaled all of the equations by around 1/10, you would end up getting the time frame that you would want. So Dr. Glazier, that's just a little update from what I had submitted last week, but otherwise the values are the same. Kind of a quick and dirty fix, not necessarily. I don't know if that's necessarily the best practice to do. I would like some insight on that.
Well, I think you could always, I mean, if you rescale time, effectively, if you multiply everything on the right-hand side by 10, that's equivalent to changing your time scale by a factor of 1/10. And so we could do it in 2 ways. You could either have used their time scale and then changed how often you're going to call it from CompuCell, because the antimony time scale and the CompuCell time scale has a multiplicative factor that's a free parameter that you can choose. Or doing what you did is fine. I think that that's a perfectly acceptable way of handling it. I was a little concerned about some of these issues, and so I think I sent you a simulation I threw together with a simpler oscillatory model. But I think what you've done there is great. I don't see any problem with that.
Okay, that's good. I wanted to I felt a little more comfortable using the antimony method just because, CompuCell makes it easy to do. But I have looked at the code that you sent.
I'm not saying you should use it. I just wanted to make sure for my own due diligence that if I'm asking a student to do something, I better be able to do it.
Right.
And since I hadn't done that one, I wanted to make sure that I had done it. So.
All right, thank you. Okay, so that brings us to this week, which is the implementation. And I am going to stop sharing my screen and open up AnnoHub. I'm not going to annotate. Sorry, I have a lot of windows open. All right. So in order to make sure that the cyclic AMP model is working how we expect it to work, I created this mini model, which I called mini chemotaxis. And basically what it does is that it seeds 9 cells, sorry. One oscillatory cell in the middle surrounded by a bunch of relay cells. Seeded that manually. Then I introduced an antimony solver, steppable, implemented the string. So then what we can do, or what I ended up doing, is that I added this model to each cell and then defined the initial cyclic AMP concentration based on how much cyclic AMP the cell contained. So those are these 2 lines. That's this line of code here. And then the way I had written the string, well, I guess what determines whether a cell is going to be relay or oscillatory is this variable A of t. So I had written the model in the default way of giving the negative value for an oscillatory cell, but then here I override that if the cell is supposed to be relay. And then here's a step function where we run the model over and over. Wait, do I have it running? Yes, wait. Yes, I do. We do. That's the very first line. Sorry, I missed it. That's this. I only have one SPML at this point. So when we open that in player, we see that the middle oscillatory cell has more cyclic AMP in its environment than the relay cells surrounding it, which is kind of what we're expecting because the oscillatory cells in the model will end up being the sources of cyclic AMP. And because the relay cells are just going to have that one spike and then kind of go to zero, we'd expect them to have less cyclic AMP. So that, and here's this, and the simulation's running. So that's my update. I think that shows the cyclic AMP is working. So the next step would be to add the cell excitability function, which won't be difficult to write because it's just like an update function that can go in a step function, and introduce an ammonia field now because that's going to affect how the cells interact with cyclic AMP. I'm going to pause that. And that is my update.
I think that looks great. TJ, do you have any comments? I know you've been talking about this.
No, I mean, both Tyler and Kali have been making excellent progress. Yeah, they're doing a great job. We have standing calls every week, and every time they show up, they have more and more done. So,
Great. I'm very impressed. It's a beautiful model, but it's not so easy to interpret. So I'm very impressed with how you're making progress on it.
Thank you.
Okay. Next I have Jeffrey.
You can share slides.
Go ahead.
So first, this is kind of, again, a test of Mechanica's fluid capabilities. There's a little object. It's actually a circle, but when I plotted it, it turned more into an oval shape. I need to fix that. And you see the flow kind of goes around it. And this is the pressure. I misunderstood what was pressure before. So now that is figured out, and you see it's higher here in the front and weaker in the back, like it should be. Here is the driven cavity with both with potential boundary conditions implemented in the top rows and with that in the bottom. And you can see that when it's not implemented, the particles kind of cluster up against the wall. But without it, it doesn't, and it doesn't. I found parameters that don't like push it too far away from the walls, so you can see it. I guess at the bottom, it's still not moving much, so it's still, I guess on the sides, it's moving a little, but the flow is much smoother with the potential boundary conditions than without it. So it seems to be working well. And then in the pressure, there's it's stronger here in this corner, which is good. Here is a video of chemical diffusion. This is a little block like a catalyst, and the particles bump into it, and you can see that they absorb some of the chemical. This is just a first kind of starting to look into this. I want to read more into SPML documentation and a little bit more on what is going on with the code before I get more involved in this. But you can see that the particles are only reacting with the catalyst and not so much with each other, which is what is good because that's what is going on in the paper. And then this is something that I was curious about now that I have the single kind of cavity working. I wondered if trying to do two would work, or if they would just kind of mash into each other. And I found some parameters for the potential that shows that you can do two cells, which is kind of what happens in the paper, but it's kind of more symmetric all around. So this is like, it makes like a torus of flow circles, I guess. And then that is all right now.
So sorry, so the drive is on the top corners? Where's the force being applied to generate the circulation in the one you just showed?
I can show it again real quick.
I know you said it last week, but I just am trying to remember.
So this isn't exactly what is going on in the paper. This is just, there is force along this wall and this wall. So this wall and this wall on this side that is driving it. But in the paper, it will be a buoyancy force driving it. So when the particles interact with this, the products of the reaction will become lighter, like this red square. They'll become lighter and move up, and then others will fill their place and react and move up. And then there will be swirls around that. So it'll look something like that still, but this is a little different, more of a brute force implementation of it.
That's fine. If you can get the contiguous flow, that's already a lot. So, very impressive.
Yeah, yeah, this is really good. Yeah, for the buoyancy force, there's still one. It's actually a really small thing that I need to add of hooking up a potential to a state variable on the object, which is it's actually a pretty simple thing to do. So I just need to do that next day or so.
Yeah. But
yeah, that's really impressive though that you got that you actually showed the difference between the plane bounce back and putting a pressure term, and actually honestly that in and of itself is a paper.
Yeah, I was.
These particle-based fluid methods are not trivial. They're pretty difficult, and so it's quite impressive.
Okay, next on my list is Fumbo.
Yeah, sure. I share my screen. Yeah, so basically, as promised, like last week, I've implemented using the mouse to do all the things. So here, in the initial state, if you click the mouse and then move the mouse, when you still clicking the mouse, like you can rotate the screen based on your mouse location. And if you release the mouse, like all the screen will just stop, like the rotate. And if you hit the reset button, it will reset the image into the initial camera state. And the next function is to shift the screen. So if you hit the shift and then move the mouse, you can shift the screen, right? You can shift the camera screen. And still, if you release the shift, it will change into the rotation state. And the last function is to use the mouse wheel to zoom in and zoom out, like that. And this even works when you run a simulation. So if you are running a simulation, you can still change the camera angles from that. And you can pause that and reset that and reset the camera angle and do it again. Yeah. So right now, I think I have already implemented all the things I think that is required to do like in this project. So in the next step, I will try to write the documentations and try to see what can be optimized in my code and try to apply this structure to the NanoHub and to see like it's the same. It has the same performance when you're applying it into the NanoHub. And I think the final step is I can combine with Jeffrey's project and try to implement his project on NanoHub using my widgets. Yeah.
Well, Andy, Vanbo, that's really impressive. I mean, Andy, you're running out of things to ask Vanbo to work on. He did it so quickly.
Yeah. I mean, that's I think. I mean, we worked together on it. Where I think it's a really great way of organizing. A team where Fanbo worked on the front end of hooking up all the widgets, and then he basically tell me, hey, I need a simulator to do this method and that method, and so now we're going to work on documenting what he's done and then packaging it up into a module where users can use it directly to build their own models. But I did actually I do regularly test on NanoHub Dev, but there definitely is some sort of it's weird on NanoHub Dev. There is a performance issue with their GPUs, and I think that's something that they have to look into, because
I think the NanoHub deployment generally would be nice, but having Mechanica working in this smoothly in Jupyter notebooks generally, you can deploy it on AWS. You could deploy it on other web services. So NanoHub or no NanoHub, this is very valuable.
Yeah.
Colab, maybe you should be trying.
Yeah, exactly. Yeah. I mean, it's weird because on the NanoHub Dev servers, compute performance is really fast. But even testing, Fanbo's Jupyter rendering code on my 5-year-old laptop, it renders much faster than on NanoHub. So, and in theory, they have like NVIDIA Teslas or something really high-end cards. So I need to work with Nick over there to try and figure out what their issue might be. But yeah. This is great. I'm really happy with this. Yeah.
Josh, go ahead.
Yep. Devin.
Yeah. So I'm just gonna talk through my project, if that's okay, and I'll explain kind of why that is in a moment. But I've implemented pretty much the whole code base that was provided in the previous CompuCell project. Did all the plugins and the additional Python functions and stuff like that. And when I try to run it, I'm getting a couple errors, and I think it might have to do with like versioning issues for CompuCell, because the old project was using like 3.7 or something older, and it's saying it can't recognize something like that now. So I'm going to have to look into that later this week. And then for next week, I'd also like to implement oxygen and possibly pH as a chemical field for the simulation, because currently it only has glucose. So yeah, that's where I'm at.
Okay, well, so please reach out to one of us to help debug,
Yes, I'll do that in the next couple of days for sure.
because Python is a great language for writing mistakes that are hard to see. And so a lot of times, I mean, I know also, I was having trouble when I was trying to do this version of the thing Kali, I had to ask, was Kali's been working on. As I said, I tried to do it on my own and I was having a bug that I couldn't find. And I showed it to TJ, and he said, "Oh, you've got your XML definition inside of another XML definition." And so there are things like that. That was on the XML side, where it's very easy to make a mistake that is obvious to somebody who's not writing the code, but it's hard to see when you're inside of it. So I'd be happy to sit down with you, or Josh, or TJ. Any of us can sit down with you and try to look at the errors and try to help you with that.
Cool. I think everyone is doing very good progress. I think James has asked your question as if this strategy was working. I think it is, in terms of like, how much progress students are getting by having this weekly meeting. That's the purpose of having these weekly meetings, just to walk through the problems as we go through the class, rather than waiting like last week to solve all of the problems at once. Okay, so let's come back to the lecture now. So last time we were talking about how diffusion in biological contexts is mediated by differences in the media that the molecules are diffusing through, and that includes cell membranes, but it also includes areas in the inside a cell that might have or throughout the tissue that might be more densely packed than other areas. We talked about how we can represent those differences in the local properties of molecular diffusion by assigning different local diffusion coefficients inside of our simulation. And to exemplify how this can be done in CompuSol, we simulated diffusion through a membrane, and we started with a completely permeable membrane, where we saw that the diffusion field, the field diffused through the membrane as it diffused just through the rest of the simulation domain, what we call the medium. And then we played with the local diffusion coefficient corresponding to the cells that compose the cell membrane. By decreasing it, we show that we went from the regime where the chemical was diffusing throughout the simulation domain equally to a domain where the chemical was diffusing more slowly through the membrane. That resulted in a different concentration profile than what we have with a completely diffusible molecule throughout the simulation domain. And then we showed also that by setting the diffusion coefficient through the membrane to 0, we can create this impermeable membrane where the diffusion molecule cannot get through. And in that case, what we showed is that with the specific boundary conditions that we shows, the concentration of the chemical field equilibrates at the concentration in the source edge. Then we decided to keep the impermeable membrane, but simulate diffusion through channels in that membrane, just as it happens, for example, with free diffusion through the cell membrane. And we showed that the diffusion profile, the concentration profile, depends on the width of the channel, and as the channel becomes bigger, the concentration looks more and more linearly, and as it gets closer, smaller and smaller, it takes much more time for the molecule to diffuse through that channel. Next, we have, up to that point, we have talked about diffusion without taking into consideration any decay of the molecules. So in terms of the biological model, all we're representing was a molecule coming out of a particular source, which was mostly an edge, and moving or diffusing through the simulation domain, either without any sort of barriers, or with membranes has barriers, or differences in diffusion coefficients in the simulation domain. But then we added a additional term to the diffusion coefficient, and this one accounts for decay of the molecule in the simulation domain. And we explained that from a biological perspective, this decay usually refers to some sort of inhibition of the molecule diffusion through, or the inhibition of the molecule itself, degradation, active degradation in the extracellular environment, rather than a spontaneous decay, which is not often seen in biological context, because biomolecules tend to be rather stable, although there's always exceptions to that. But the idea is that we can now represent not just the supply of the molecule, but we can, coming from a particular source at the borders, but that we can also represent the consumption of the decay of that molecule as it diffuses through the tissue. We mentioned that that the fact that these molecules decay through tissue establish what is known as a characteristic diffusion length for the molecule, and this diffusion length has important biological consequences. For example, the distance between capillaries is determined by the diffusion length of oxygen, such that the capillaries must be at a distance such that the oxygen can diffuse through the tissue and get to cells before decaying below a certain threshold. And this is also true about the length of how many hepatocytes you will have in a certain in the liver, in a certain tube in the liver, and also it determines the length of propagation of signals during embryonic development. Then we explained that when we account for this diffusion, when we account for this decay, we go from a regime where the gradients are, or the concentrations decrease linearly from the sources, to concentration profiles that decay exponentially from the source. And the rate of decay of that that concentration is given by this term, the square root of the decay rate over the diffusion length of the diffusion coefficient. And then we call the diffusion length the square root of the diffusion coefficient over the decay rate. Then we implemented this diffusion with decay in Compusol. This is quite straightforward to implement. We have this global diffusion coefficient in the XML that specifies what the diffusion coefficient is for the molecule across the simulation domain. And then we have this global decay constant that it specifies what the decay rate for the molecule is across the simulation domain. And we show that when we set that global decay constant, as I explained before, the concentration profile decays linearly from the source. And when we include the decay, the concentration profile decreases exponentially from the source. And then the concentration profiles look quite different. Then we talked about secretion and exocytosis. We talked about how these are important processes that cells use to release intracellular substances that might be signaling molecules, or might be excretion from different cellular processes. But we mentioned that from our perspective, from the perspective of modeling, all we care about is that the secretion of a certain molecule has the net effect of increasing the concentration of that molecule in the extracellular environment over the cell. And we talked about how that can be implemented using a simple secretion plugin in the CC3D-ML file, and we have a couple of options as to how can secretion be done. We can secrete at a constant rate. We can secrete when the cell is in contact with another cell of a specific cell type, or we can force the cell to have a constant concentration over each of the pixels that belong to that cell. We also talked about how we could also specify secretion using Python, and in that case, this gives us much more control over how and when secretion happens, and actually whether the secretion varies in time as a function of any other parameter that we might want to make secretion a function of. And that we have these options that we can secrete it with respect to the cell. We have different options as to how we want secretion to be implemented. Secretion at the boundary, secretion at the center of mass, and then if it's at the boundary, whether it's inside of the boundary or outside of the boundary, or whether we want it to be secreted over every single pixel of the cell. And then we showed how this looks, how each of these secretion options look like when they are implemented. And in this particular case, if we look at this screenshot here, you guys can see that the maximum of the field is 1. And in that case, it's 1 because I set it to be a secretion as constant concentration. And that what that does is that every single pixel of the cell always has a maximum a concentration of 1. And then from those pixels outward, the molecule diffuses such that the maximum concentration will always be 1, the ones given at the pixel of the cell. In this other example, I set it up to be secretion at a constant rate. And in that case, the maximum is not given by the, it's given, it would be what the maximum value of the field in the simulation domain would be is given by the secretion rate over how much I'm taking out, either at the borders or by decay. And then we also explore how the different options of secretions, how other options of secretion look like. In this case, secretion is happening inside the cell boundary, which is the yellow line here. And you see that the secretion, the pixels right in the inside of the cell boundary are the pixels that have the highest concentration of the field, and that is because the secretion is happening on those boundary pixels. In this particular case, is outside of the cell, and in this third example here, it's at the center of mass of the cell. The center of mass is off a little bit because there's not a central pixel to the cell, so it shows one of the 4s that are at the center of mass of the cell or nearby the center of mass of the cell. We also finished the class by talking about the opposite, uptake. And we mentioned that, that if secretion has the net effect of increasing the concentration of the field over the pixels that correspond to the cell, the uptake has the opposite net effect. It decreases the concentration of the field over the pixels corresponding to the cell. But in this case, since the concentration cannot go below 0, right, because the molecule, the cell could take all of the molecules are at outside of let's say they're outside of the membrane, but the concentration, the number of molecules outside the cell cannot go negative. So our model of uptake needs to reflect the fact that the concentration cannot go negative in any part of the simulation domain. So that means that our model of uptake needs to be saturated by the concentration, such that the uptake goes to 0 as the concentration of the field goes to concentration of the field in the vicinity of the cell goes to 0. And we explained that the way Compusol implements that is that the user specifies 2 parameters. The user specified what the maximum uptake is and specifies what the relative uptake is. And what happens is that when the concentration goes below what the maximum uptake is, let's start this way. When the concentration is above what the max uptake is, the cell takes whatever the maximum uptake is. Let's say we specify that the maximum uptake is of units 2, whatever units we decide to assign to it, and the concentration in the field is 4, then at that particular time step, the cell will take 2 out of the field that is 4 in its domain, right? But if the concentration is below the maximum uptake, right? If the concentration of the field is below 2 in this example, then the cell takes a relative uptake, a relative amount. So that it would be a fraction of 2 that we specified as the relative uptake rate. And this is to ensure that when the concentration goes to 0, as you're taking just a fraction of it, the concentration outside of the cell never goes negative. Uptake in CompuCell, it's specified used in Python, and we looked at the code to do so. We need to call this get field secretor object that refers to the field that we specified as the field that the cells is uptaking from. And then we need to specify then the arguments of, we call this uptake function on that secretor. And we have also the same, we have similar options to those that we had with the secretion in terms of where on the cell the uptake is happening. But then this uptake function takes 2 arguments. It takes 3 arguments. It takes the cell that it's uptaking. It takes what the maximum uptake is, and then what the relative uptake is, just as I explained before. And here we see an example of what this code looks like when implemented in the Python step. Also keep in mind that when this uptake inside this uptake function returns an object that has many attributes, but if you want to call or refer to the amount that it was uptaken by the cell, then you need to reference the specific attribute tod or property tod underscore amount, and that gives you the number. So if in one step the cell took 2, then this rest that tod amount is 2, whereas this res is really an object. It's not the number that you care about. And this is important because we'll do it. We'll have an exercise now where we implement this uptake. I did not show this last time because I'm not sure. I don't think we got to actually implement the uptake. But here is an example of what the uptake looks like when it's implemented under different conditions. So in this particular condition, I had medium as secreting the field at a constant value. So I set the constant value in medium to be 1. And then I set the uptake inside the cell to happen on each of the pixels of the cell. And then what you see when you implement it that way is that the field remains constant throughout every of the pixels that correspond to medium, and it decays in this concave way when inside the cell as the cell uptakes from the field. So you can imagine that it's sort of like falling down like a hole or a sink. In these 2 cases, I had a different initial condition or boundary condition. I said instead of having the medium secrete, I had a constant value at the edges of the simulation domain. And then I had this molecule diffuse inward and then being uptaken by the cell at the center. and then the concentration profiles look quite different, right? And then when you compare it to, or when you change the uptake rate, the maximum uptake rate and the relative uptake rate, what you see is that in these two different cases, the molecule diffuses, the concentration profile changes because the how much of the constant of the molecule is being uptaken by the cell changes from one simulation to the next. Quick question: in which of this is the cell taking more of the chemical field? Any guesses? You can sort of look at these plots too, even though they look similar in their shape, the scales are different. So this goes all the way to 0.2 and this is 0.8. So which these two scenarios would you think the uptake rate is higher?
The middle plot?
Right, because the concentration decreases more as you get closer to the cell, and that is because the cell is taking more out of its environment. Okay, so just a quick reminder, we will be working just like last week. I might ask people to pull their codes and show and walk. So we will walk through with the codes with you. So just be prepared to present your code and volunteer to work through the exercises as well. So now that we have, now that we can leverage what we've learned in previous lectures to combine cell growth with nutrient availability. And before we modulated, we have experience modulating cell growth and proliferation using cell pressure, right? And in that particular example, what we cared about was that the lack of space was the mechanism inhibiting cell proliferation and growth. But now we can take this a little bit further, and we can use our chemical field as a nutrient that is available to cells so they can use for cell growth and proliferation. And what we know about how the biology works is that as cells undergo, as cells progress through the cell cycle, they have to synthesize more proteins, right? So they consume more energy. Their respiration increases, such that they can duplicate their genetic material and some of the proteins that they need for cell division. So when those resources, metabolic resources, are not available to the cell cannot undergo cell division, and they usually stay at a quiescent type of state where they are arrested in their cell cycle. And if the availability of those resources falls below a certain threshold, then cells can actually die due to lack of metabolic resources. And we will implement examples of this in our example of these three types of cell proliferation states in our next example. But this is actually something that is observed in tumor spheroids, and And it's also observed in avascular tumors in vivo, where the proliferation of cells, where cells at the outer surface of the tumor are consume the available nutrients, and the cells in the interior of the tumor are deprived of those resources, so they die. And then cells in the middle, there is usually a middle layer of cells that are arrested in their proliferation because the resources available to them have fallen beneath a critical threshold, but not enough to kill the cell. So the cell has enough resources to maintain their basal metabolism, but not enough resources to proliferate. And in theory, there is nothing that prevent us. Oh, so before, the way we will do this, right, is that we will modify our growth rate with a function of whatever our uptake is from the field, right? And the same way we do this with the same way we did with the pressure, where we modify the how fast the cell was growing by how much pressure the cell was experiencing due to the difference it's in target and actual volume. So now what we will do is that we will modify the growth rate by how much of the resources that the cell is taking up. And there is this caveat here that nothing prevents us from actually combining multiple of these inhibition mechanisms together, because in reality, these mechanisms operate in combination as well. There is inhibition due to lack of available space, but there is also combination inhibition due to lack of metabolic resources. And in fact, those are usually also correlated. So in this first example, we will implement an example or a model of diffusion limited growth. And I'm gonna walk through this first to the implementation with you, but I'm gonna ask someone to actually run the simulation once we're done implementing it. So the first thing we're gonna do is that we're gonna start to edit. And we are going to, just give me 1 second. And we're gonna create a simulation with 100 by 100 lattice. Let's call it the growth.
Are we using the blob or rectangular initializer when it says uniform initializer?
Oh, it's a blob initializer. I'm sorry. Right. If you don't wait for me, like you guys can move ahead if I'm going too slow. 100 by 100, and it's actually the blob initializer.
Josh, do you want to be showing your screen as well as?
Oh, I'm sorry. Oh, thank you for reminding me. I'm I thought I was actually showing my screen. Do you guys see my screen now? My whole screen?
Yeah, now we see it.
Okay, I'm sorry. I'm sorry about that. Yeah. Let's periodic here on the X and Y axis. No flux in the Z axis. Let's initialize one cell and one field. using the diffusion solver FE. And then we're gonna need cell growth, cell division, contact, and volume local flex. And we need the secretion as well. Yeah, we need the secretion as well. Let me 8.1. I'm sorry, I'm taking notes so I can update the slides as well. We're not gonna care about any of this stuff right now. Right. Right. So in the secretion, we need the cell type, we need medium, we need constant concentration to be 1. And what this is gonna do is that it's gonna set the constant concentration in medium to be 1, just as I showed before. Okay, finish the simulation. And then more. Okay. So so all of this is fine. Now we need to, the secretion data is, as I've said before, the secretion data is always, when you load the plugin, the secretion plugin, the secretion plugin is loaded independently in XML, and then it has the secretion data inside of it. But then that secretion data is going to be repeated again inside the diffusion solver steppable. And I prefer deleting it from one of them so as to ensure that I don't have to modify the data in both of them if I make any changes. So I usually delete it out of the secretion plugin. Then we're gonna set the diffusion constant to be 0.1 and the decay constant to be 0. And the reason we want that global decay constant to be 0 is because we want any uptake out of the environment of the diffusion field to be due only to the uptake of the cell. And we do the same in this diffusion specific data. Another option we can do, because the diffusion data will always include the global diffusion information and local diffusion information that you can determine by cell type. But what we will do is that we can either set it the same, or we can delete that information that is not necessary, because we're not going to specify specific diffusion coefficients and decay constants by cell type. Okay. Next, since we set the concentration of the field to be 1 due to secretion by medium, this information as well is also loaded. The secretion data, it's loaded automatically. As I said, in the secretion data inside the steppable, just make sure to comment it out or modify it as you need. And it's not gonna be secretion, it's gonna be constant value, constant concentration. I'm just gonna type it. So what's important to notice here is that when you load the Compusol project, there is a lot of stuff that is loaded by default in the XML. And even though you have specified certain things in the wizard, you always have to come back to the XML and the Python step to make sure that what you specified in the wizard is what is written in the XML, and that you modify any information as needed. So we don't need, since we specified the concentration on the medium to be constant, we don't need to specify values at the edges. And so what we will turn them into periodic boundary conditions. And then we need to initialize a single cell in the center of the simulation domain. There you go. So that would initialize a single cell in the center of the simulation domain. So we have set up everything that we needed in the XML file. Now with the Python stepable, so if I launch my simulation now, what I should see is a single cell in the center of the simulation domain. And if I look at the field, I should be able to see that the field is 1 everywhere where there's medium. So that's my cell in the center of the simulation domain. And it's 1 everywhere where there is medium. 0 in the center of the cell because that cell is not medium, so that's not how it's initialized. Okay, now what I need to do is to modify my growth rate such that the growth rate is a function of how much my cell is taking from the environment. Let's start by having our cells just grow at a constant rate. We've done that before. And then if we run our simulation, okay, that crashed. One second. By the way, I uploaded a finished version of this code in the Canvas. So if you guys go to Canvas and go to demos and go to lecture 8, you'll find a version of this code that is actually running. So I'm not sure what the error was in that one, but just to ensure that I'm not having any just to ensure that I'm not having any typos, I'm gonna look at the code that I've generated. That's not that one. Okay. So if you look at that simulation, in the steppable functions, what we'll see is that I have defined this, defined this salt dictionary item, growth rate, that I'm gonna use to track how the growth rate is being updated by how much the salt is uptaken from the environment. And then I have already implemented different options as to how to modify that rate. But the first thing we will do is that we will have them, just as a demonstration, we will have them grow without limitation, right? And that's just gonna it's just gonna produce what we've seen before in terms of cells growing and occupying the whole simulation space. Okay. Let's make this a little bit smaller, 100 by 100, and put it in the middle, or let's make it 200 by 200. So, and you guys will see why we were playing with the size of the simulation domain at the end of this simulation, this example. Okay, let's relaunch the simulation. And let's open the graphic window so you guys, okay. So now the growth rate is gonna be, right, it's gonna be unlimited, and I'm not gonna look at the field right now because I have already uptake implemented, but the idea is that if you run this simulation, you're not modifying what the salt is taking up from the environment. So the growth rate is just gonna be as if there was no limiting factor. So it's gonna eventually fill up the whole simulation domain, right? So what we want to do now is that we want to modify that uptake, that growth rate, by how much the cell takes out from the environment. The way to do that is to call our secretion function, our uptake function. And the way to call the uptake function is we go to the CC3D Python and we go to secretion uptake, and then we call, depending on which of the options we're interested in, but in this particular case, we want to take over every single pixel of the cell, so we call uptake inside cell. And notice, this is important, if we want to be able to grab and reference how much the cell is uptaking, we need to add this total count to the function. So it's gonna be uptake inside cell total count. And now notice that I need to specify again what the max uptake is and what the relative uptake is. And I said in the instructions that you should define them to be 0.05 for the max uptake, and then 0.025 for the relative uptake. And then when defining the function, since I am taking over every pixel of the cell, and all I want the cell as a cell to take out of the field is 0.05. But if I just type 0.05 here, that means that every pixel of the cell will take 0.05. But what I really want is for the cell to take 0.05 over its whole domain. So I have to divide that max uptake by the cell volume. That's what I do in that case. And now we can play again the simulation to see how the field changes as the cell fills up the whole simulation domain. Okay, my computer crashed. My NanoHub crashed, not sure why. Who's willing to take over from here so we can walk through what the code looks like, what the diffusion limited growth looks like? Anyone?
I can do it.
Okay, who? That's Fambo.
Yeah. But I may switch my computer, and I will join soon after 30 seconds.
Okay, do take the screen though. Don't take away the screen. Oh, well, did he say that he had to relog? Because I think we lost him. Oh no, he's here.
Yeah, so I will share the screen right now.
Okay. Right. So yeah, so right. This is all good. Right. Just don't modify the growth rate yet, 44 by the apps by the uptaking amount. Just so coming out, right. So we can just see how the cell is uptaking from the field, even though the field is not modifying the growth rate of the cell. Okay. Right. So
You have to run that.
Yeah, run the simulation now. And I'm gonna go for the next exercises. I'm sorry, I'm gonna go to my local version of CompuSol to avoid further crashes. I'm not exactly sure what's happening with my NanoHub. Right. So there we can see so right. Okay. You can see that the
I think this may be some problem about my initializer.
Right. I'm not sure. Yeah, this is fine, but I'm not sure. Do you initialize the volume constraint and the contact energies? In the XML file. Yeah, but you will see it in the XML files. Right, in the XML files, right, you do have the volume constraint. And you do have the, okay. Yeah, it should be working.
Here, you mean the volume constraint?
The volume plugin, right? And you have the contact energies as well.
Can you point like which line do I have the rolling constraint?
I think you're fine. Run the simulation again. Right. Right. So the cell is gonna keep growing and it's gonna divide because we have set up we still have the constant growth rate, but we have set it up such that the cell is actually taking up from the chemical field now, right? So what's gonna happen is, as you can see, as the cell divides, it creates a gradient from the outside to the interior of the blob. And this is exactly what we wanted to see, right? And since we have no decay, right, we set it up at the very beginning, this gradient is solely due to how much the cells are uptaking from their environment. Okay, so now pause this simulation or stop it, right? And now let's go back so we can actually modify the growth rate now by the how much the cell is taking up. Right. So now we're modifying. So now what we're doing here in line 44 is that we are multiplying the growth rate by how much the cell is taking, given how much it could take, right? If the cell is taking as much as it can, then the growth rate is gonna be 1. But as the amount that the cell takes decreases, then the growth rate should decrease as well. And then right in line 45, Fambo, can you put just the term after the start, the absolute over the max uptake? Right. Yep, this is fine. And then go up just to make sure, because the purpose of adding this dictionary entry is that now we can visualize the growth rate inside the cell blob, right? And that is initialized here in line 24. You were already working on it, so that's fine. Save this simulation now and we can run it. You can hit and maximize the window and then we can add a new window, here to display the growth rate. Right. Okay, now you can play the simulation. Okay, and then what we should see is that the cells start growing, but as they start taking up from the environment, their growth rate should decrease in a way that resembles how the gradient is decreasing in as the field gradient is decreasing as well. Right, and you can see it here that as the cell grows, then the growth rate of each independent cell decreases as well. As let's think of it as a tumor. As the tumor gets bigger and bigger, and you guys can see that these cells at the edge, they are proliferating as fast as they can, whereas these cells in the center of the blob are decreasing, are proliferating or are growing slower and slower. Okay, any questions so far about this? Okay, cool. I'm gonna take the screen back, Fambo. Thank you so much. Thank you for having the code ready. Not sure what's happening with my NanoHub, but what I want to show here is I want to show how we can modify that growth rate to account right. So this is what we saw. I'm sorry. So this is what we saw, right? As the cell increases, and then if we make the simulation domain bigger and bigger, what will happen is that we will have a domain of cells that the proliferate as fast as they can. Then we have a domain of cells that are proliferating slower and slower. And then we have these cells in the center that have a growth rate that is 0. Right. But in this case, our cells are not really dying. What's happening to these cells in the center is that they stop proliferating. So they acquire that quiescent proliferative state that we talked about. We could also simulate, and this is probably something I'll leave for, I think it was in the homework already, in the homework that you guys are submitting today, and I'm not gonna stop to simulate it now. I'll just show the code really quick. But we can also simulate cell death by modifying the way in which cells are the way in which cells the way in which cells grow. So here I have different options. And in this particular case, I have defined this basal metabolism that I call 0.2. That is basically specifying what is the uptake that the cell needs for its survival. And then if the uptake goes below that level, then what happens is that, let's say that this uptake here goes below 0.2, then what happens is that the growth rate becomes negative. Any guesses as to what happens is if the growth rate becomes negative? Because so far we've played with increasing the target volume all the time as the simulation steps go by, as the simulation progresses. Any ideas what would happen if the target volume decreases as the simulation progresses?
With the cells here.
Right. So that is a way of simulating cell death. If we simulate cell growth and proliferation by increasing the target volume, we simulate cell death by decreasing its target volume. And this particular function will ensure that if the uptake is above a particular level, we get increase in the target volume. But if the uptake is below a critical threshold, then we get decrease in target volume, and that would simulate cell death. And we can think of more complicated more complicated models of cell death. This is just a zeroth order approximation, right? Because in this particular case, let's say a cell starts decreasing, but the concentration over it suddenly increases, then the cells will stop decreasing and start proliferating again or growing again. Whereas in reality, once cells activate their apoptotic or pathways, those are usually non-reversibles in the sense that cells will continue going through, even if the conditions that trigger those pathways change. Okay, does that make any sense? Cool. So what you would do if you get to implement these models and you play with the diffusion coefficient is that you get this type of branching of your cellular blobs. And these branchings can be thought of, and James shared a paper recently with me that uses these branching properties as a way of simulating how malignant tumors can infiltrate nearby tissue. And the reason this branching is happening is because cells are penalized for not having enough if not supply of nutrients. And since the supply of nutrient is coming from the medium, then cells are trying to maximize their contact with medium, and that's where the branching happens. And with appropriate parameter values, you would get this branching to happen to get more or less of this branching to emerge from the parameters of the model. Okay. I did wanted to say that the function that we use to modify cell growth in the previous exercise is quite simple. Basically, the growth rate is modified by a linear function of how much the cell is uptaking. But there are more complicated functions that we can also use to simulate the same effect if we wanted to account for the fact that cells can take at, depending on the availability of its resources, cells can take at maximum rates and so on and so on. But just as an example, that function gives us what we wanted to in terms of having a growth rate that is a function of how much availability of resources there are. Now we will continue building on this idea of chemical fields and how cells respond to them. And one of the many different responses that cells can have to chemical fields is what is known as chemotaxis or chemokinesis. And in this case, cells respond to chemical concentrations, either nutrients, oxygen, or other signaling molecules, and move through the gradients of these molecules. There are other physical factors that might also induce cell movement. But in general, we will represent cell movement due to certain, let's say, field using the idea of chemotaxis, the idea that the cell moves because it's able to sense difference in the gradients of these molecules. The mechanisms by which cells move through gradients are actually quite complex, and they vary from cell type to cell type and from organism to organism. But in general, the two types of response can be classified in chemokinesis and chemotaxis. So the chemokinesis is usually the main way in which bacteria will move, and in this case, it combines it is a combination of some sort of directed persistent movement through a up or down a gradient, right? Because not always cells do not always move up a gradient, right? There might actually be a chemorepulsant that makes cells move in the direction that minimizes the concentration. But in the case of chemokinesis, what the bacteria does is that it has a characteristic persistent length in which it moves in a certain direction for a certain amount of time, and then it tumbles and changes orientation and direction. The way it does, and the reason it does it is because that gives the bacterium the ability of explore the space in a more complete way, and to find areas of, let's say, their environment in which the chemoattractant might be in higher concentrations, but they might be unable to detect because of their local gradients. The way they do this is by using their flagella. And if you guys take a class in biophysics, you will spend certain amount of time calculating the energy that the cell requires to move these flagella. But the idea again is that the cell uses this flagella for directional movement, in which it moves in a certain direction up or down a gradient, but it also uses it for tumbling and changing their direction in a random direction. We will play, if we get to it today, one of the simulations today that we will play with sort of simulates or models this type of bacterial movement. Eukaryotic motility, as always, is more complicated, and it usually includes certain flagella, cilia, but also phylopodial extensions and lead edge formation, because cells can polarize in a certain direction and change their polarization due to exposure of certain gradients, and they can move their and depolymerize their cytoskeleton to extend in certain directions. Here is an example of a amoeba that is moving in due to certain detection, right, by extending their by leading edge formation. There's also other examples of another classic example of chemotaxis is the Dictyostelium amoeba that responds to cyclic AMP, and this is what Callie was talking about earlier. Here's a couple of videos that explain how that demonstrate how the Dictyostelium responds to this gradients, and actually chemotax can form certain patterns. I don't know. Yeah. Yeah. For some reason, this one's not playing. The links are gonna be posted here. Even if the video is not playing right now, yeah, I will reference it to you guys so you can actually watch them by yourselves. But they're quite interesting to see how the cell reacts to a pipette that adds cyclic AMP the solution or the media, and how the cells polarize and move in that direction. James, do you want to add, because this model is quite interesting.
So
but
you couldn't play that culmination. Which one did you want? Which?
The only one I was able to play was this one.
Well, that shows the culmination phase at the end.
Right, right. So
I don't know why it's showing it as an inset though, rather than showing the whole. I think you need to find a different version of the same movie.
Right.
Somehow anyway, that's too bad that that's not. I suppose you could just you could exit the slideshow and just play them in your browser for a second.
Okay. 1 second.
I don't think you should take too much time, but it's useful to see them.
Let me see. Yeah. Okay, let me share my screen again. So James, you might want to explain what's happening here with this specific organism, or Callie, for that matter, since she's working on a model of dictyostelium.
Well, when dictyostelium are in starved conditions, they will aggregate together to form a slug. That's what all the literature seems to call it. So they're all joined together, and what will happen then is they will, through chemotaxis, they'll move towards areas of higher cyclic AMP concentration. And then that's here. So this is like the fruiting body's a little bulb part at the end. And this is showing how it's kind of growing into it's a stalking amoeba, so that the cells will differentiate and form different components of the stalk, and will culminate into this fruiting body, which is what I'm trying to model, fingers crossed.
Cool.
So what's happening is that, again, it's a little bit surprising. So when the cells starve, they send out a signal, and instead of running away from the place where they're starving, they come together. When they do that, you see them flowing together here. And then when they do that, they pile up to form this mound. You don't really see the migrating slug in this video. There's another video you had where it showed the slug migration. And then in culmination, spores are cells that have shut themselves down in an orderly way and built a hard surface wall to protect them against unfavorable environments. And what you see there is that some of the cells form spores, and some of the cells die and become rigid and form that stalk. And the idea is that then, if some animal or the wind knocks the spores off, they can distribute and move far away, because the cells normally live in the ground. They can't move very far away, but if the local environment is, there's this slug growing. If the local environment is unfavorable, they want to move far away. And so the way they do that is by sporulation, and then moving out vertically out of the ground, so that they can be blown away by the wind or moved by an animal. And the issue that the slides are bringing up is that the cells that form the stalk die. And of course, as multicellular organisms, we're used to the idea that the disposable soma, namely that really the only things that matter are our sperm and eggs. The rest of us is disposable. But these are organisms that live as single cells. And so it's not clear why one single cell would sacrifice itself for another single cell. But of course, if the cells that are nearby to each other are related because they divided from a common ancestor, then there can be selection for these kinds of collective behaviors. And so this particular organism is often used as a model of the origin of multicellularity, because unlike us, where we're mostly multicellular except during reproduction, these are organisms that are mostly single cellular, except when the situation is bad and they have to disperse. And so chemotaxis is one of the key things that is involved here, because they are responding to chemical signals to aggregate. And actually, when they're crawling, they're also responding to chemical signals, and the culmination itself is due to chemotactic motion. Okay.
Cool. So, yep. So let's go back here. So yeah, this highlights the highlights the importance of chemotaxis in driving several important cellular processes. In terms of how to model chemotaxis, the basic idea for modeling chemotaxis is that the cell movement needs to be driven by the gradient of the field in the cell surface, right? The cell has to be able to somehow sense that the concentration changes from its, let's say, edge to its tail or around its membrane, and then move in the direction that increases the concentration in the case of a chemoattractant, or decreases the concentration in the case of a chemorepulsant. And then this proportionality term is that what we will call lambda chemotaxis, and that lambda chemotaxis scales how the force experienced by the cell to move in the direction of the gradient. It scales the force to the concentration gradient. So this simple model has some limitations that we will come back to and try to solve later. So for example, if the gradient is very steep, right? Let's say like in an exponentially decaying gradient, then the force would be very, very big. But the cells have a limit as how fast they can move. So the force experienced by cells needs somehow to be saturated or need to reach a maximum, such that the cell reaches a maximum speed due to the chemotaxis. And there are other aspects related to the diffusion that we will not care about right now, but we can come back to it later. The idea is that the way chemotaxis is implemented in CompuCell is slightly complicated, but if you guys have questions, we could either come back to this at the end of the class, or in this lecture at the end, there's reference slides that explain step by step how the chemotaxis is implemented. But again, the idea is that the concentration of the field is measured at two different voxel pixels, and then whether the cell moves in the direction of the concentration increase depends on this energy term that we will call the chemotaxis effective energy term. And again, it's determined, this chemotaxis energy term is going to be determined more or less by the lambda chemotaxis and the gradient of the chemical field. Notice that if the lambda chemotaxis is negative, then we are talking about a chemo-rich pulsing because the cell will move in the opposite direction of the gradient. If the chemotaxis is positive, then we're talking about a chemoattractant, and the cells will move up the of the chemical gradient. Oh, and this is very, very important because usually these changes in concentration over the simulation domain are small. If we actually want cells to be able to move due to the chemotaxis effective energy, the lambda chemotaxis usually are very large. And we will see examples of this in our next exercise. So the simplest way of specifying chemotaxis in CompuCell is to do it through the XML. And we do this by loading the chemotaxis plugin. And once we load the chemotaxis plugin, we need to specify what the cell type is that it's moving due to the gradient and what is the field that is causing the cell to chemotax, and then what is the associated lambda chemotaxis. So let's do that. Let's just implement a simple chemotaxis on a linear gradient. I'll walk with you on this one from the beginning so you guys can see how the chemotaxis plugin looks like. But again, I'll ask someone to show the code when we get to run the simulation. Let me share my screen, my whole screen, and initiate the poll. Okay, so what we will do is that we will create a new simulation. Let's call it Chemotaxis simple. And then we want a 200 by 200 lattice, periodic boundaries on the X and Y axis. We want a blob initializer so we can, oh, I'm sorry, it's not a 200 by 200. It's a 200 on the X axis, but 100 on the Y axis. And then, so we want a rectangle. And then we want a blob initializer so we can initiate one cell. Let's just add a single cell type. And let's do something fun. Let's call it dictine. And then let's add a cyclic AMP field using the diffusion solver FE. Okay, and then all we need is we need chemotaxis, we need contact, and then we need volume flex, not the local flex. So since we have enabled chemotaxis, when we hit next, we will get this new plugin that we haven't seen before. And then this plugin, we need to specify what the chemotaxis type is. We will explain this later, what the saturation terms are doing. But for now, the regular is what I've been calling here simple. And then the field that is going to cause my cell to chemotax, the cell type that is chemotaxing, and then the lambda chemotaxis. Let's set that one to 500. And then this specifies through which cell types my chemotactic cell is going to move through. Okay. So now what we will do is we will set a linear gradient, and by now we should sort of like be experts in doing that. So what we would do is that for linear gradients, we cannot have any decay. We do not want any diffusion coefficients by cell type. We do not want any secretion. Then what we want is our minimum, our value to be, our boundary conditions to be constant value on the x-axis, right? We want 1 on the left edge and 0 on the right edge. And then we want periodic boundary conditions on the y domain, along the y-axis. And here is an important, so let me modify. So an important, if we initialize this with these particular conditions, we will have to wait for the diffusion field to reach, we will have to wait certain amount of time for the diffusion field to reach equilibrium. But if we know how to, for this particular case, we know already how to solve the diffusion equation. And then what we can do is that we can set the initial concentration to be the same as the equilibrium concentration. And that is given by this equation. Right? Because when x is 0, I want it to be 1. When x is 200, I want it to be 0. And then I want it to decrease linearly between both edges. So that's going to give me my initial concentration to be the same as the equilibrium concentration. I'm sorry, I'm going a little bit fast. Let me, look at the poll. Going a little bit fast because we've talked already about how to create these linear gradients. Then what we will do is that we will create a cell, a single cell in the center of the X domain and the Y domain. And then if I execute this simulation, I was actually meant to ask someone to do it. So who's with me that wants to run the simulation? It's not extremely complicated, what we've done so far. Last thing is that we want to plot the position of the center of mass of the cell as a function of time. So to add any plot data, we come to our step-level function, and then in the store function, we use scientific plots setup. We call it position versus time. This is position. And then let's use the one that it's dots. This one. And let's call this data series POS for position. Then in my step function, all I need to do is to, as I iterate through the cell, I just have one single cell. I need the center of mass on my cell on the x-axis because my cell is only gonna move on the direction of the gradient. Or at least that's the only one I cared. Any movement that occurs along the y-axis, it's gonna be due only to membrane fluctuations and not due to the chemotaxis experienced by the cell. So what I need to do is I need to collect somehow what the position of the center of mass of my cell is in the X domain. And to do that, I can come to CC3D Python, cell attributes, center of mass. And you guys see that you already loaded the center of mass in both X, Y, and in all the X, Y, and Z positions. But again, all I care about is the X position at this point. And then once I've collected the center of mass, I can add a data point that is going to be that center of mass. Okay. And that's all we need to complete this exercise. So who wants to show their code? Two people are with me, so.
Is there some problem specifically that people have that where we can help them?
Right. I was also going, as I said, I was going super fast over certain stuff because specifically over creating the linear gradient because we did that last week. But again, the idea is that you set the values to be one on one edge of the simulation domain, 0 on the other edge. And then for the initial concentration, we know what the linear gradient is, the linear profile is, so we can set our initial concentration to be that. So we don't have to wait for the field to equilibrate. Okay, 3 people are with me, so at least one should be able to show their code.
I can do it.
Okay.
Okay. I think I'm most like 90% caught up to you.
Okay, so first, is this your simulation? No, you're not running the same. Is that the same simulation?
Yeah, this is just my
Oh, but not the one you're executing, right?
not yet.
Okay, so you can delete line 73 because we don't want the dict T cell to secrete anything. Right. Everything else is fine. Go up to see what the pod just go up to see what the pod parameters are. Good. And then chemotaxis. Go to the chemotaxis plugin that 500, and then go to the bottom so we can see what the initialization is. The radius, set it to 5. Yeah, that's fine. And then did you add the plotting?
In the separables?
Yeah.
Not yet.
Okay. Here, add the setup. Right. Here, yeah. Just change the data set here to call it pause in line 20 and make it dots. And then get rid of line 21. And then right in line 32, get the, right. Right. But then now you need the center of mass and the X direction, which is X, COM with cell that, right, because it's a property of the cell. X and then cap without the dot between X and COM, right? And then take this and put it inside the cell loop. That's it. There you go. So the idea again is that you're collecting what there is an indentation error that you're gonna get.
Oh yeah.
There you go. The idea is that you're collecting what the center of mass of the cell is, and then plotting. There you go. Okay, and then can you display the field as well? Right, and the reason you're not seeing your field is because you'll have to update the settings. So if you go to configurations here, fields, and then the fix is the maximum is gonna be 1. We know that because we set it up to be 1, right? Another option is just to let it flow. So you guys see that it's already equilibrated when you started the simulation. And then can someone tell me what they see both in the plot and the simulation? What? So Tyler, you told me, what are you seeing?
I see the center of mass is, or is it, is it the center of mass is moving to the left?
Right, right. And that is due because that is because the cell is responding to the gradient, linear gradient that we created. Okay, so if you see here, what is the slope of this curve telling you? If this is position, right, on the y-axis, and this is time, what is the slope of this curve telling you?
It's a speed then, if it's position over time.
Right, right. It's a position over time, right? It's telling you what the speed, how fast the cell is moving through or to the left, right? This is important. This is gonna come in the homework. So please do notice that the speed is relatively constant, right? So the cell with certain fluctuations that are due to the membrane fluctuation. So now you have a combination of two cell movements. You have a cell movement that is due to the cell membrane fluctuations, and then you have a movement that is due to the chemotaxis. Then what you're seeing here is a combination of those two movements, but you see that the cell, even though there is a combination, even though there is random fluctuations associated to those membrane fluctuations, the cell is still moving at a constant rate from right to left, and that is due to the chemotaxis. Now let's do something for me, Tyler. Go to the chemotaxis XML, and then increase the lambda chemotaxis to 5,000. And run the simulation again. Okay, so what's happening now?
The slope is steeper.
The slope is steeper, which means that the cell is moving faster, right? And also notice that there is way less fluctuations as well, right? The contribution of the cell movement due to chemotaxis overtakes the movement due to the membrane fluctuations, and then you see less variability in how the cell moves throughout the gradient or through the gradient. So what happens when the cell reaches the border, right? Is we set the borders to be periodic, but the cells is detecting that on the other side of the border, the field is lower, so it's not moving on that direction. It's basically staying at the position where the field is at maximum. Okay, does that make sense? And we will come back. You will explore a little bit of of this in the homework. Okay. Tyler, can I take back the screen from you? Okay. So yeah. So if we initial as the cell is gonna move in the direction that increases the concentration as it detects that gradient, and then the speed at which it moves is proportional to that lambda chemotaxis. And the response is linear on the gradient, which means that if the gradient is linear, then the velocity at which the cell moves across the simulation domain is going to be constant. Okay, and those things are important. So we also have the option, we have, by specifying, and this is a recurrent theme, and by using Python specifications, we will have better control as to what we do with some of the model parameters, and then we're also able to change them dynamically. And we've seen that with increasing the target volume, right? If we specify it in XML, then we are unable to change it in the Python step, and we are unable to make our cells grow. Same is with chemotaxis. If we specify our chemotaxis in the XML, then we cannot modify it in Python. But there is also the option of modifying our lambda chemotaxis in Python, and we'll see later what we will use that for. But at least here, what we want to show is that what the syntax is to do this modification. First, we call this Chemotaxis plugin add chemotaxis data. We need to do this every time we initialize a cell that we want. Every time as we initialize a cell and we want that cell to be able to respond to the field, we need to call this function. So we add the chemotaxis data to the cell corresponding to the field, and then we set the lambda for that field. So that would be the equivalent of specifying what the lambda chemotaxis is in the XML. But then we can access and modify what the lambda chemotaxis is by calling this anywhere else in the code. We need to call the get chemotaxis data. And then this reference CD, the CD object, we can call the method get lambda, and that will give us the value of the lambda chemotaxis. And then again, we can use the set lambda to set a new value of the lambda chemotaxis. So in this particular case, we grab the chemotaxis data, we grab the value of the lambda chemotaxis, we decrease it by 3, and then we set it as a new lambda chemotaxis. Okay, so this gives us more control over how to specify the chemotaxis, and if we want to be able to change it dynamically as the simulation runs. So this is very useful if we want to specify some dependency. We want to evaluate the dependency of the chemotaxis forwards on some of the simulation parameters. And what I'll do with this one is, because this one is a little bit more complicated, I will just show you what we did in the code. But the idea is that the basic idea of the code is that we will set at ordinal value of the lambda chemotaxis to be 500, and then we will have a small script that is gonna increase this lambda chemotaxis at this rate, at the rate of 1.2. anytime one of these conditions is met, either 1,000 Monte Carlo step is reached, or every time the cell reaches the edges of the simulation. And remember that by changing the sign of the lambda chemotaxis, we can turn our field, we can make our cell become a chemoattractant or a chemorepulsant. Okay, and then what we will do is that we will, as we would change this lambda, we will record the velocity of the cell, right, by measuring the slope, because we know the cell reacts linearly to the gradient. And then we will plot what the velocity is as a function of increasing the lambda chemotaxis. Okay, and this is going to become more obvious as I showed in my code. So, and again, this code is available to you on it's available to you on Canvas. So nothing changed in this simulation, except that I commented out the specifications of my chemotaxis. I could, right, in the chemotaxis plugin in the XML, I commented out any data that I had inside of it, because I'm gonna specify it using Python. Initial conditions are the same that I said on the previous simulation. And then I have the position versus time plot that we just showed, but I also added a new plot window that is gonna plot what the velocity is as a function of lambda. Okay, that's nothing terribly new there, but here is the important part. So see how this is in the start function. And once I have initialized my cell, I call the Chemotaxis plugin add chemotaxis data. And the way I can access that code is by coming to CC3D Python, chemotaxis, by say cell ID define. And then it gives me the code snippet. All I need to do is use that code snippet to specify what the field is going to be that my cell is going to move to. In this case, it's called field, and what the lambda chemotaxis is. So again, this is the equivalent of initializing this information in the XML file. Okay. I've also created 2 additional cell dictionary entries that are gonna that I'm gonna use to calculate the velocity of the cell. And then what I do is that, so these are the conditions that I just specified, right? I said if every time 1,000 Monte Carlo simulation steps go pass, or the cell reaches one of the edges of the simulation, what I do is I change the direction of my lambda chemotaxis and I increase it. So what my cell would do is it will go on one direction until it reaches the edge or 1,000 Monte Carlo step pass, and then it will switch direction until the condition is met again. And then it will keep doing it. And then I will record the velocity of my cell as it goes, as I increase the lambda chemotaxis. Okay. I don't wanna spend a lot of time in this code. If people want to go over the code in more detail later, I would be happy to discuss it. But what is important here is because what I really want you guys to see is how the lambda chemotaxis controls what the velocity of the cell is. So I did something here. Okay, no, that's not it. It's here. Okay, no, that crashed. I think, yeah. Right, all of this is fine. Let's launch it again. Line 52. Okay, so I think I need to specify what the chemical, right. I need to specify, when I delete, this is important. When I delete the information from the Chemotaxis table, I need to leave what the field name is that it's gonna, what the field name is that it's gonna be operating on the chemotaxis. Otherwise, I'm basically enabling chemotaxis toward that fields. But if I delete that information, then I get an error. But again, I initialize my cell in the center of the simulation domain. My field is equilibrated from the beginning, and I started with a lambda chemotaxis of 500. What's going to happen is that the cell is going to move, and then it's going to change direction and increase its lambda chemotaxis. So we can see here how the cell is responding to the chemical field. And as soon as it reaches 1,000, it increased its lambda, but it also changed direction. So it's basically doing this at the moment. And then in this direction, what I do here is that every time the cell changes direction, then I use this data here to calculate the velocity. So you can see the velocity here, and then I'm plotting the velocity as I increase my lambda. And then you can see the cell starting to move and change directions. The reason I did not want to spend a lot of time on the code itself is because you can type the conditions yourself, but the idea, again, is that you want the cell to be able to switch directions and increase the lambda chemotaxis. Okay, I'm gonna pause here for a second. What are you guys seeing in terms of the velocity as I increase lambda chemotaxis? Which is this plot here. So I've been increasing lambda, and what happens to the velocity of my cell as I increase lambda?
It increases as well.
Right, it increases as well. And you can tell like up here that it's increasing quite linearly, at least in this regime. And let's run it for a little bit longer. So now you guys see that as it reaches the border, it changes lambda. It starts moving in the opposite direction, and the lambda increases as well. Okay. So let's let it run for a little bit longer. So what's happening now? So in this regime here, the velocity increased linearly with the increase in lambda chemotaxis. But what's happening when I got here?
Is it staying constant?
I'm sorry, say that again.
Is it staying constant?
Right. It reached a maximum speed that the cells can move at. Right. It cannot, it's oscillating around 0.25, and these 0.25 are pixels per Monte Carlo step. And James, do you want to explain why is that the case? Why do we reach this
Sure.
speed limit?
Well, if you think about what's happening in Monte Carlo step, each time you're picking a random site in the lattice and asking, does the cell move forward by 1 pixel? And in principle, each point on the surface of the cell is visited once per Monte Carlo step. And that means that unless you're accidentally lucky and the same position of the boundary of the cell is visited twice in a Monte Carlo step by the randomness of the order in which you visit, the cell can't move more than 1 pixel per Monte Carlo step, because each point of the boundary can only move 1 pixel per Monte Carlo step. In practice, you can't move even that fast. Because you don't have a guarantee that the order in which you'll visit the pixels is a particular order, and you actually pick the pixels at random. So during one Monte Carlo step, you pick as many sites as there are lattice sites, but you're not guaranteed to visit every site. And so in practice, you can't move as fast as 1 pixel per Monte Carlo step. You could only move about 1/4 of that. But there's an absolute limit, which is 1 pixel per Monte Carlo step. There's a practical limit, which is about 1/4 of that. And you see that very dramatically here.
Another thing to notice is that as we increase this lambda, notice that this has been plotted on a log scale because we're increasing that lambda exponentially. But the cell starts deformating quite a lot. And that is because the force that the cells experience, the effective force that the cells experiences to move in one or the other direction is quite high. And as we'll see in the next exercise, that force can be high enough that it makes the cell disappear. But in this case, what we wanted to show is that as we change this lambda and we keep increasing it, we increase more or less linearly. There's a regime at the beginning that is not so linear, but more or less linearly until we reach this saturation at the effective speed limit at which cells can move in CompuCell, which is 0.25 pixels per Monte Carlo step. Okay, so it's important to notice that this code can be reused to test for the dependency on other parameters. I have just 10 minutes left, but as a homework, we'll ask you to change some of these other parameters like the cell size and the contact energy, and whether you have more than one cell or not, to test whether to see how the speed or the velocity of the cell changes as a function of changing those parameters. What is important to notice to keep in mind for the homework is that you can see that you should use for the specific gradient that we are using here, that Lambda chemotaxis of about 1,000 or 10,000 is good enough to give you one second, to keep you in this linear regime. All right. And then what you will do is that instead of changing Lambda chemotaxis, then you can change other parameters like the target volume of the cell and see how that changes the chemotaxis. Okay, so let's come back here. So the other problem that we will experience, as we were pointing out, is that if the gradient, the simple chemotaxis term that we are using assumes that the cells are going to respond linearly to the concentration difference. But this is a problem when the concentration gradient is exponentially increasing or decreasing, because then the response itself will increase exponentially. And then what that would lead is to cells disappearing because the force that they are experiencing is too high. And in reality, cells do not go as fast as they can, as you cannot just increase the cell and expect the cell to move, increase the gradient and expect the cell to respond always linearly to the gradient, to the change in the gradient. But there is a saturation or a speed limit that the cell would experience, even if the gradient increases above a certain threshold. And to control for these factors, we can adapt the Lambda chemotaxis such that the cell stops responding. Such that if the gradient increases exponentially, the cells do not respond exponentially to the change in the gradient. I have 9 minutes, so I'm gonna try to showcase the next couple of simulations, then we can come back to them next class. But the way we do this adaptation is by dividing the Lambda chemotaxis by 1 plus what the concentration of the cell is at the center of mass. So in this particular example, instead of using the linear gradient, we can generate an exponential gradient using the same simulation we had before. Let's do it quickly. The same simulation we had before. The simulation where we created the linear gradient. So in that one, when we want the linear gradient, we set our decay constant to 0 and we initialize it at the equilibrium concentration. If we want the exponential gradient, then we can set the global decay constant to be non-zero, right? And the equilibrium concentration now is given by this equation, not by the linear equation, right? Because the gradient now decays exponentially. We talked about that last class. But then what we do now is that instead of having the cell chemotax linearly, we modify the lambda chemotaxis by the value of the field at the center of mass of the cell. So if we look at this particular example, we first loaded the chemotaxis data and notice that in this case, we have to set the lambda chemotaxis to be a very, very huge number, 1e to the 30. And then what we do in the step function is that we measure the value of the field, right? The value of the field at the center of mass of the cell. And then we adapt the chemotaxis by the value of that concentration. And then when we run this code, what we see is that if we do not have this adaptation, the cell responds exponentially to the exponentially increasing field to the point where it disappears when it reaches very, very steep gradients near the edge of the simulation domain, because the force exerted over this is too big. If we use this adaptation term instead, even though the gradient is decreasing linearly, what we observe is that even though the field is decreasing exponentially, what we see is that the response, the chemotaxis response, is linear in that gradient. And the reason is because as the gradient increases linearly, the decreases is normalized by the value of that concentration field.
And it's worth saying perhaps that kind of response is quite common in biological systems, what's called perfect adaptation. Here we're seeing it in the context of chemotaxis, but it actually happens, for example, in vision. When the light levels go down, the sensitivity of your photoreceptors goes up. Clearly, it's not perfect in the sense that if it gets dim enough, you can't see very well. But one of the things that you have to deal with, for example, in chemotaxis, is that the chemical concentration might differ by a factor of 1,000,000 from one place to another. And if you want to measure a gradient, if you are optimized for measuring a gradient for a very large concentration, then you don't see a small change at all at low concentrations. And if you are optimized for seeing small changes in concentration, then you're saturated and completely clobbered by high concentrations. And so what you see is with dark adaptation in your eyes is that you actually change the number of receptors in response to the concentration. If the concentration is high, the number of receptors goes down. If the concentration is low, the number of receptors is increased. Now, it takes time for that to happen. Here, we're assuming that response is instant. But in many cases, that response is pretty fast compared to the other things that are going on. And so making the assumption that you have perfect adaptation is actually not bad.
Right. And it also preserves the stability of the simulation, right? Because in this particular case, cells do not disappear just because they are exposed to a exponentially increasing gradient. Another thing to mention here is, and you'll see it if you play with this code that is also available to you, is that this term is gonna determine how far in the gradient the cells get to. If you make it 10 times smaller, the cell might get up to here instead of all the way through the end of the gradient, like in this particular example. And maybe that's something to play with in the homework. So before we go, in the next 3 minutes, let me show really quick how this chemotaxis can be used to model or simulate cool biological phenomena. So we could use this to simulate a bacterial movement, as we talked before, in which we provide a constant supply that it does not diffuses, and it's just uptaken by the cell or disappears because of the cell trajectory. And potentially we could measure that persistent time before the tumbling, which was what we're talking about in terms of cell movement. And we could explore what are the parameters that determine how the length of that persistence in one direction, right? You can see the tumbling happening here, and then the persistent movement happening in this direction. And another very cool example is we can model, we can pretty closely recreate this macrophage chasing the bacterium movie that is pretty well known. And in this particular case, what we do is that these cells are here just to represent obstacles and to represent these blood cells. But then we initialize a bacterium that secretes a field that attracts the macrophage. And then we set the contact energies between the bacterium and the macrophage such that the macrophage repels the bacteria. And there is also other components that we can set. For example, we could have our macrophage actually eat the bacteria, which is not happening in this particular simulation. And we could modulate what the response is of the macrophage to the signal secreted by the bacteria. Does it absorb it or not? And that has a particular effect on the simulation, or whether it responds linearly or as a adapted chemotaxis, as we explained before, and whether that has an effect or not on the simulation. We will probably come back to this at the beginning of next class.